So welcome to Chapter 3. We're going to jump right into the nucleic acid part of the chapter, and then the subsequent casts will be about proteins and then enzymes. So we'll finish off our classification of macromolecules, first with nucleic acids. We'll talk about the different nucleotides, different kinds of polymers, and then we'll move on to proteins. You've already seen some of these structures because last week we did the sorting of the molecules activity. You were able to see different bases, the nitrogenous bases. We've identified the difference between ribose and deoxyribose, the presence or absence of that OH group there. If you put these two together, you get what's called a nucleoside. And then if you combine a nucleoside with a phosphate, you get what's called a nucleotide, which is the monomer or the building block of DNA and RNA. The thing that makes the nucleotide different, or the nucleoside different, would be the different kinds of bases that are attached up here. These orange boxes are these things down here. There's two classes of bases. There's ones called pyrimidines, the cytosine, the thymine, and then if you're talking about RNA, thymine is replaced with uracil. But similar structures, alternating double and single bonds, carbons and nitrogens. Whenever you have nitrogens and oxygens around, these are polar, and their electronegativities are different between the different elements, and then you might be having some ability to hydrogen bond. The other class are called purines, the adenine and the guanine, or the A and the G. These are a double ring, again alternating double and single bonds, the capacity to have some hydrogen bonding going on as well. Unfortunately, you do want to memorize all the names on this page. One thing to think about is the pyrimidines have a Y here. So do the cytosine and the thymine. Again, uracil is just a replacement for thymine, so that's a different thing to memorize on its own. I also tend to remember that the pyrimidines are the single rings. It's a backwards relationship, so longer word, single ring. Purine is a shorter word, double ring. Usually helps me. So you can build these into the polymer with our condensation reaction that we talked about in the other chapter. So the place where you get to remove the water is the H that's attached to the phosphate, and then the OH, which is attached to the sugar. So these numbers in red are actually important as well. The carbons, remembered, are numbered 1, 2, 3, 4, and 5. So on the fifth carbon is a phosphate, and on the third carbon of the chain that's already growing is the OH. So it's said that the polymer grows in the 5' to 3' direction. So you're always adding the 5' OH or phosphate to the 3' of the sugar. Have the condensation reaction take place, and then here's our polymer, again growing 5' to 3', 5' to 3', and so on. Here's our 3' OH ready to get another one if it was coming on here. So this is ribose. You can see the OH is here. And then another thing to that nomenclature is that this is called the phosphate sugar backbone, or sugar phosphate backbone. And it kind of will be usually depicted as sort of a band or ribbon, and then the bases are hanging off sort of in the middle. So what are some differences between DNA and RNA besides the sugar we've been talking about? We know ribose and deoxyribose. The bases it uses are different. RNA uses A, C, G, and U, and it makes a single strand when it puts it together into a polymer. And DNA is deoxyribose, and it has all the bases except the uracil switched out with the thymine. And DNA tends to make a double strand, and you probably heard about the double helix. We'll talk about it in a few minutes. So how could DNA make a double helix, and then also how RNA makes some structures, is basically a result of hydrogen bonding between these bases. So they've got sort of an abbreviated picture here. We've got our sugar on the side, and then focusing really on our pyrimidines and our purines. Purine and pyrimidine on this side. I'm sorry, purine and pyrimidine on this side. We see that the A's and the T's like to make 2 hydrogen bonds, and the G's and the C's will make 3 hydrogen bonds. Because of the way they are oriented, this is the type of bonding they always do. And because they can make hydrogen bonds, you can make these sort of hairpin loops in the RNA molecule. So RNA is usually made as always made as a single strand, but it likes to make hydrogen bonds. See, there's a U and an A binding together, a C and a G making a hydrogen bond, and that holds it together in these funny little structures. We're going to see RNA create a structure like this and help regulate gene expression. We're going to see RNA fold up like this and make tRNA or transfer RNA. It can make all different structures and function both as sort of an enzyme as well as an information molecule. So then DNA, we've always heard about having the double strands. And let me just orient yourself here. Here's one strand moving in the 5' to 3' direction. Here's our sugar, phosphate sugar, phosphate sugar, phosphate sugar, so on, backbone. And then the bases are hanging off into the center. You can see that T binding with the A, G binding with the C. On the other side, 5' to 3', going in the anti-parallel direction, carefully positioning it so that we can make all these hydrogen bonds. One thing to remember with it that's really cool about the purines binding the pyrimidines, the same distance is always saved between that those two strands, and that allows your DNA molecule to make this perfect double helix, and it winds up like this. You've probably seen pictures of this. So the ribbon, the blue ribbon, is the sugar phosphate backbone. They color-coded the bases hanging off in the middle. The A is always with the T, the G is always with the C, and see the guanine is the purine, it's the double ring with the single ring, so we always preserve that same distance between the ribbons. We'll talk later about who proposed the structure and all the experiments that were done around it. And then also, hopefully this week we're going to have some time to purify DNA from strawberries so you can see what DNA looks like in a test tube. Lastly, why do we have DNA? Basically, it's an information molecule. When it's a polymer of all those different letters, the A, G, C's, and T's, it can be organized into different patterns. You could think of A, G, C, and T as sort of the letters, and they can make up words. And then the words can make up to bigger things in proteins. DNA has two roles. Basically, it's a blueprint to copy itself. So it's there to be replicated and to be preserved to pass on to the next generation. And then it's also used as a template for gene expression. So the DNA gets transcribed into another molecule, RNA, which leaves the nucleus, goes into the cytoplasm for the translation when a ribosome finds it and makes it into a polypeptide or a protein. The RNA or the DNA is coded in the RNA, and it basically codes for the exact sequence of those proteins that are going to get made. Some other words that are related with DNA and thinking about how it's an information molecule. The complete set of DNA in a living organism is the genome, so you can think of that as every single base in there. Those bases are organized into groups called genes, and those genes can be transcribed into RNA and then ultimately usually made into a protein. Not every gene is always made into a protein. Sometimes they have other roles, and we'll talk about those as time goes on. And not all DNA in the genome is really encoded in a gene. Some is structural. We'll talk about how those work later. Really cool thing about DNA is that it is a blueprint for its own copy. In later chapters, we'll talk about the experiments where they did to prove that DNA copied itself in this way, that one strand is preserved and a new one binds to it. And then DNA, like I just said, is a template for making different RNAs, which we'll make into different proteins. So it's a coding information. And then lastly, we'll talk about this when we get to chapter 16, about how the DNA bases that have been conserved over time can show how related organisms are in evolution. Now that they can sequence whole genomes, we're really understanding in a more clear way how organisms are related to each other through history, and maybe what came from what and how related they are. It'll be the holy grail for figuring out ultimately who's related to who.